Good evening, good evening, good evening, or good afternoon, I should say, because it's still a beautiful afternoon here, and I want to welcome you. My name's Andrew Petter, president of Simon Fraser University, and let me start by acknowledging that we are, of course, privileged to be gathered on the traditional unceded territories of the Tsleil-Waututh, Squamish, Coquitlam, and Musqueam peoples. And it's a real pleasure to welcome you to the President's Faculty Lecture with Professor Angela Brooks Wilson. I think this is the first time we've actually held the President's Faculty Lecture in recent years right here on the Burnaby campus. And I said, you know, if we hold it up here in Burnaby, will people come up to attend a faculty lecture that's really got the community in mind? And you're proving that you have. So thank you very much for being here. Hopefully some of you are aware that SFU has very much made it our vision to be Canada's engaged university. We believe that we can add more to the community by reaching out and connecting to the broader community in ways that traditional ivory tower universities haven't done in the past. And we think that the university community, our students and faculty and staff, gain a lot of benefit back from that relationship. So whether it's local or whether it's global, we really believe in engagement as a means of both sharing our knowledge and gaining value and knowledge back from the community. The President's Faculty Lecture Series, which is part of SFU Public Square, is very much a signature initiative of that vision and that commitment to be an engaged university. By providing you with opportunities to hear from time to time and to engage with SFU's leading research faculty, these lectures are very much designed to enlighten and promote dialogue on issues that are important to you, issues of public interest, but issues that are also occupying the time and energies and talents of our research faculty. Now, there will be a chance for you to raise questions or offer comments after the lecture, and you're also welcome to continue the conversation at a modest reception that we, modest not in the richness of the conversation, but in the richness of the food that is being offered. But you're welcome to stay back, and our speaker has very kindly agreed to stay back and spend some time chatting with those of you who wish to chat with her. I'll also either warn or encourage you by the fact that this lecture is being filmed and will be available on our SFU YouTube channel. So should you ask a question in this forum, you may end up on the internet. So whether that's an inducement or a caution, you'll have to decide. Now let me introduce the faculty member who we're very privileged to have as a speaker for this afternoon's lecture. Dr. Angela Brooks Wilson is professor and chair of our Department of Biomedical Physiology and Kinesiology at Simon Fraser University. She's also a distinguished scientist at Canada's Michael Smith Genome Sciences Centre and at the BC Cancer Research Centre, where she leads a cancer genetics laboratory. She's a member of the Canadian Cancer Society's Advisory Committee on Research. She's also an alumnus of Simon Fraser University. She studied her Bachelor of Science in Biochemistry at SFU, went on to study her Master's of Science in Medical Biophysics at the University of Toronto, and earned her PhD in Genetics at UBC. Her current research focuses on the genetics of susceptibility to cancer, particularly blood cancers in families and populations, and on the genetics of healthy aging. And of course, healthy aging is very much the topic of her discussion today. She's published widely in these areas, participated in numerous studies, and those studies are helping to advance our understanding of cancer and of aging and many other issues as well. She's leading the Healthy Aging or Super Seniors Study, which you're going to hear about today. And I had the privilege of hearing her on the radio today. I understand she's been doing a lot of media over the last 2 days, so hopefully she has some voice left for us. And with that, we'll soon find out. Please join with me in providing a very warm welcome to Dr. Angela Brooks Wilson. Dr. Brooks Wilson.
Thank you very much, President Petter. It's a real pleasure to speak today. I have a bunch of friends in the audience. I can see a bunch of former students in the audience, and you're a friendly-looking bunch of youngsters. Actually, I really enjoy talking about the super seniors study. It's nice to talk about some very sort of positive things. But let's see. Okay. So I'm going to go through quite a lot today. So fasten your seatbelts. I'm going to tell you a little bit about our motivation for the study, a little bit about human genetics in 5 minutes or less, super seniors, and I'm going to go through 4 selected research hypotheses. How many of you in the audience are scientists? Okay, about a third of you, I guess. So for everyone else, hypotheses, or when my graduate students destroy my hypotheses, is more like it. That's how science is done. I'm going to take you through about 4 of those. And throughout the presentation, I'm going to highlight the work of students, particularly SFU students. The motivation for this study is actually comes from the other work going on in my lab. And this is a family, Cancer Family 133. Sounds a little bit impersonal, but this is for privacy. Cancer Family 133 is the largest family in our lymphoma susceptibility study. And to give you a feel for what we work on, and for those of you who are not used to looking at pedigrees, the circles are women, the squares are men, and where the symbols are completely blacked in, they're affected with a lymphoid cancer. Just ignore the red dots. That's our code for we have a blood sample. And the unshaded ones are unaffected. You can see that in this very large set of brothers and sisters, 3 of the brothers have Hodgkin lymphoma. And they developed Hodgkin lymphoma in early life or midlife. Their aunt also developed lymphoma, and a cousin developed lymphoma. So they clearly have a strong family predisposition to lymphoid cancer. We don't know what's causing it yet. But looking at families like this made us think about how we could ask other questions that might be relevant to the question of why do people get cancer and why do people get other diseases. At one point we asked ourselves, why don't we look at super healthy people and ask, why are they healthy? Why are they so unusually healthy? So we began a study of healthy aging. We're studying healthy aging in part because age is the major risk factor for almost all common chronic diseases. So the risk of everything, almost everything, increases as we get older, and in part due to increased exposure. And understanding healthy aging may actually give us insights at the biological level that might allow us to design interventions, not just lifestyle changes, but actual drugs or other interventions to mimic that state in people who don't have the most favorable genes. So human genetics in 5 minutes or less. You'll have to tell me afterward if we succeeded in teaching it in 5 minutes or less. You're now an expert in looking at pedigrees. So here's another one. This pedigree is a hypothetical pedigree made up to illustrate a point. And again, the circles are women, the squares are men, and the dark symbols are people who are affected with some disease. You can see that the great-grandfather here had a disease which he passed to both of his children and half of his grandchildren and half of his great-grandchildren. So this is a very straightforward pattern of transmission. He passes the gene on to his kids, and everyone who inherits the gene is affected. Actually, in real life, it hardly ever happens this way, fortunately. What usually happens is we get situations like this. The same pedigree structure, the same affected great-grandfather, but now half of his children are affected, and only half of his grandchildren are affected, and both of his great-grandchildren are affected. So you can see from the fact that the disease is showing up in the great-grandson that it passed through these 2 unaffected people. So why weren't they affected? In genetics, we call this penetrance. And the penetrance of a genetic variant or a genetic disease is hardly ever, I would almost say never, fully penetrant. So just because you inherit a disease-causing variant doesn't mean you'll develop disease. And in my opinion, one of the key questions in human genetics is, what is it about these people who carry a disease gene and yet don't develop a disease that lets them evade the disease despite their genetic predisposition? So this is the sort of thinking that we bring to the Healthy Aging Project as well. Here's another little bit of genetics lesson for you. As you know, we have 2 copies of every chromosome, and every chromosome is double-stranded. So just look at the top strand here and the top strand here. This is a type of genetic variant called a single nucleotide polymorphism. And in this case, and a nucleotide is the A, C, T, or G of the genetic code. On this chromosome, there's a C on this strand. And on the other chromosome, this could be the chromosome from the mother, and on the chromosome from the father, there's a T instead. So there's a spelling difference on this chromosome. And this is actually an excerpt I've taken from the actual human genome data. This is an important cancer gene, and the green highlights show various, we call them polymorphisms, multiple forms throughout the genome. So to illustrate a point of how important genetic variation is in everything about us, our phenotypes, our various sort of how we manifest externally what we're like, our disease predisposition. I chose 2 human specimens completely at random to illustrate this point. These are really nice human specimens. So they actually show, and this is true, about 1 in 1,000 of the base pairs of the DNA sequence of these 2 gentlemen would differ. And given the size of the human genome, that means they will have about 3 million differences between them. So they have 3 million differences, and they look different. In some ways, they look kind of similar. But the current challenge of human genetics is entirely to correlate those 3 million differences with all the differences in phenotype that we see in human populations. No small task. And to top off that task, another principle is that environment comes in as well. So, for example, longevity and healthy aging, which are different things. Longevity is how long you live. Healthy aging is whether you got there in good shape. So healthy aging and longevity are both what we call complex phenotypes, meaning that they're affected by both environment and lifestyle, and they're affected by genetics. So I'm a geneticist, and I'm studying healthy aging. How much of healthy aging do you think is genetic? Would anybody like to guess? Parveen? Oh, we've had somebody say 30%. So you are absolutely right. Okay. So some say 25 to 30% is about it. So I actually study the small part of healthy aging, and I do that because my expertise is genetics. But we've done a little bit of work on the environment as well. So now I'm going to tell you about the phenotype we're interested in, which is the super seniors. And super seniors are individuals who are 85 or older. We chose the age 85 because it's a gerontological term for oldest old. It's a category in gerontology. We then defined the super senior phenotype as being individuals who have never been diagnosed with cancer, except non-melanoma skin cancer, cardiovascular disease of any kind, major pulmonary disease except asthma, diabetes, or dementia. So they are people who are 85 or older and they don't have these big 5 diseases. And we chose those diseases in part because we wanted to study a phenotype that's not only desirable for the individuals, but also for the healthcare system. If we could have more older people who don't have those big 5 diseases, we would also be saving a lot of money. And that is important in very practical ways as well. So I'm going to introduce you to a couple of super seniors, real super seniors. Many of you have heard of Dal Richards, who was quite proud to be one of our super seniors. He would participate in events and put on charity events. At one point, they were attended by a group of super seniors who were thrilled to hear the big band sound of Dal Richards at a charity event, and really impressed me with how quickly they could eat desserts, which might answer one of the questions you're probably going to ask me later, and which I found very reassuring. So Dal died about a year and a bit ago at the age of 97. Robert Wiener is now 107 years old, and he exercises every day. When I speak with this gentleman, I realize I should be exercising more. So he literally exercises every day, and he's relentless. He told me he has had hockey injuries from his youth, and that they prevent him from lifting his arms straight up over his head. So what he focuses on is exercise bike, because he can exercise with the bottom half of his body without worrying about the arm limitation. So these are two super seniors. Robert is interesting in part because his brother was also in the study, and his brother was just short of 110 years old when he passed away. And the odds of that are having two brothers of these ages in the same family is 1 in about 2 million. So Robert is he's trying to break his brother's record. He seriously is, and we wish him well. So the objective of the super senior study is to identify genes and genetic variants involved in long-term good health and potentially resistance to cancer and other diseases. Remember the concept of penetrance. And I sort of apologize for this slide. I actually designed it to show the work to politicians. Actually, I shouldn't make fun of politicians, but this slide illustrates the study design, which is a case-control design. And so we invite as many super seniors as possible to participate in the study. We compare them, in our case, to a control group that is middle-aged individuals. Everyone asks me, why do I compare them to middle-aged individuals instead of less healthy older people? And there's a reason for that. It would be asking a different kind of question. If you arrive at the age of 85, you're already pretty fabulous. And to compare to a person who's 85 and even healthier might not be a big enough difference to be easy to detect experimentally. And so I compare to the midlife group quite deliberately, because the midlife group is a group that's collected at an age before all the bad stuff starts, before the health problems. Many of these diseases and the mortality that's associated with them starts about age 50 and continues until later. So I'm really comparing an unselected group to an elite group. And that's our study design. Recruitment of participants took many years. We've recruited them in 2 phases. We're continuing to recruit. Initially, we collected the first super seniors from Ministry of Health lists. So after appropriate Research Ethics Board approval, we were given a list of random people over 85, which we contacted. And we found that about 1 to 2% of people were actually qualified for the study. So we ran through our list, we ran out of lists, and then the rules changed, and we were not able to get lists anymore. And so we turned to other methods. We approached a group called BC Stats, which has access to the ICBC driver's license database, and we asked them to search for seniors who were over 85 who had renewed their driver's license successfully in the last 4 years. They contacted those people, asked them if it was okay to give their name to us, and we were able to recruit about 100 people that way. And the rest have been from press coverage. So because the supers are an elite group, we're able to take volunteers, however they may come to our attention. The midlife control group must be absolutely randomly collected. And so this is only through Ministry of Health lists. I can't take volunteers for the midlife controls because the volunteers would probably be interested in health and therefore might be atypical and might skew our results. So the super seniors, I think I've explained all of this. Everyone gave informed consent. And one of the things we did when interviewing the super seniors was we had them bring any drugs they were taking out on the kitchen table during their interview, and the interviewer would write down the drugs they were on, and we would exclude people who actually had a disease that was specifically for a drug that was for one of the big 5 diseases. We did quite detailed health status assessments, taking family history, for example, and we did blood pressure measurements and geriatric tests of function, and took a blood sample for the DNA analysis. So I think what most people want to know about is the characteristics and the lifestyle of the super seniors. This actually was not a study which was designed to look at lifestyle. If you were going to look at lifestyle in particular, the best way to design a study is to recruit a bunch of people when they're young, follow them over their entire life, and ask them, what are you doing all along the way? That way your data is current and it's very accurate. In contrast, we recruited elderly people and then we said, what have you done all your life? Okay, it's going to be subject to some recall issues, so we have to take this with a grain of salt. But here are the basic characteristics of our phase 1 super seniors. The average age was 88 and a half. The average age of the controls was 46 and a half. The average, this is phase 1. Robert Wiener was in phase 2, so he's not in this group. The oldest was 105. The median year of birth was 1916. And body mass index is a little bit interesting for the super seniors. Their body mass index, and normal body mass index is 18.5 to 24.9. They were on average at the upper limit. Can we see this here? 24.5 is their average body mass index, which means they're at the upper limit of the healthy body mass. So most of them are not skinny. A few of them are skinny. And I find this incredibly reassuring. Some of them are actually in the obese range. Now, I don't think you should go run amok and start eating pastries right away. But it just shows that, at least in some cases, being in the obese range is still compatible with going on to be a super senior. So very, very reassuring. For lifestyles, we were very impressed to find that about 3/4 of the super seniors are physically active. Now, we define this very broadly. If you're doing something, lawn bowling, walking, raking leaves, this sort of thing, anything was counted as being physically active. But the same definition was applied to the controls, and about the same proportion of them are active. We tried to do a detailed physical activity questionnaire of the super seniors and ask them how much activity and what types they did in every decade of their life. And sadly for us, the conclusion of that study was we could not measure it. It was just too incomplete. And so we have to be content with kind of an approximate answer here. Smoking is also, as we might expect, hardly any super seniors smoke now that they're over the age of 85. Very few. And anecdotally, the ones who do smoke very little in quantity. The heavy smokers basically don't get to be super seniors. That's my interpretation, and that's what I would expect. About 11% of the controls were current smokers. But about half of the super seniors were past smokers. When you think of this, they lived their youth at the time of World War I, when smoking was actually promoted. And so a lot of them started, but then a lot of them quit later on. And some of them smoked up to 20, 25 pack years, so non-trivial amounts. They quit early enough that they were still able to go on and become super seniors. So I like to say that that's an optimistic message that it's never too late to quit. Also very, very reassuring is that 72% of them drink. I have a little bias here. So most of them drink, but when you look at the average, 3 drinks per week is the average, and that includes the numbers from the non-drinkers. So the drinkers are going to be a little higher. The range, it was actually quite wide, from 0 to 28 drinks a week. That's pretty high, but keep in mind, we don't know how long that person was doing that. So maybe that was a temporary thing. And another theme, the controls are, again, wider in range. I think I forgot to tell you, the BMI for the controls goes up to 48.4. So the controls are much, much wider in range, and the same thing with the drinking, 0 to 41 drinks per week. We used gerontological tests to characterize how fit and how cognitively capable the super seniors are. And we found, based on these standard gerontological tests, timed up and go, they were fast in this test, showing they have high physical function. On the mini mental state exam, they had high scores, showing high cognitive capacity. On a geriatric depression scale, they had low scores, showing low levels of depression. And on a study called the Instrumental Activities of Daily Life, things you are capable of doing around the house, they also showed, most of them, very high function. Now, part of this, things like low depression, may be because we do have an ascertainment bias in our study. We're taking volunteers. The volunteers may be more engaged. They may be more interested, more willing to be interviewed in their home by an interviewer. And so there's a certain level of non-randomness to them. So we do have to interpret our results in that sense. Most exciting to me as a geneticist is that the parents of the super seniors, based on the super seniors reporting how old their parents were when they died, the parents lived a decade longer than their contemporaries. A decade. This floored me. I would have expected it would be about 3 or 4 years, but it was 9.9 years. So if you think of this, if the average year birth of the super seniors is 1916, the parents were probably born in the 1890s or maybe even earlier. And in the 1890s, living the mothers lived to an average of 79.3 years and the fathers to 74.5 years. That lifespan, that average lifespan for somebody born in the late 1800s is quite remarkable. It's not all of them, though. So they didn't all have long-lived parents. Some of them had both parents die young. Some of them had one long, one short. Some of them had parents who died in the big Spanish flu epidemic in the early part of the last century. So it really was very variable. We did a phase 2 recruitment about 2 years ago. We have 200 more super seniors. So the data I've described was from the phase 1, the first 500. The next 200 have been interviewed and the data is being analyzed. And our 2 interviewers were 2 SFU students. Monique Sikon is a student leader and health sciences student who did our interviews in Victoria. And Olivia Aguiar is a famous SFU soccer player, student athlete, and a BPK major. And she did the interviews in Vancouver. So I'm going to go into 4 little vignettes, 4 hypotheses. And I'm going to tell you about telomeres, personality, exome sequencing, and I'm going to talk about one gene, because if I try to talk about more than one gene, you'll be here all night. But first, I would like to show you this picture. This lady is Olga Kotelko, and she was a super senior. Olga is a famous masters track athlete and a really remarkable lady. She passed away at the age of 94 a couple of years ago. And she had so many gold medals, about 400 of them, if I remember seriously, that she would give them to her friends just for mementos. And no, Olga did not win those medals because she was the only one left alive in her age category. That was not the case. She would run circles around 70-somethings. She actually found it kind of annoying that if there was nobody in her age group, she had to be put in the younger ones and beat them. So she was really remarkable. So the first study was on telomeres. Telomeres are structures, repeating structures at the ends of chromosomes that protect chromosome integrity. It's kind of like they're usually compared to the little plastic things on the ends of shoelaces. And as people age, their telomeres naturally shorten, so, and plus or minus a certain amount. So our hypothesis was the super seniors have telomeres that are long for their age. So how many of you agree with the hypothesis? What do you think? Okay, one of our faculty members. Okay, hands again. Looks like about half of you. Okay. Well, you're wrong. And I was wrong too. I thought that hypothesis was very logical, but my postdoc, Julius Halshek-Wiener, proved that it was wrong. And this is a curve which is shows how, for a population, the percentile distribution of telomere length with age. And you see the super seniors here, and they're just about right on the 50th percentile here. So the telomeres are not long for their age. But if you look carefully, you'll see that they are actually more tightly distributed. So they are more tightly clustered around a median than the control telomeres are. And this was statistically significant, and we saw this in multiple blood cell types. And it was quite a surprise. And so I usually like to say that they have Goldilocks telomeres. Not too long, not too short, but just right. Seriously, we now call this the Goldilocks hypothesis. And we don't have an answer for this yet, but we recovered from the demise of our first hypothesis and just moved on to another one. So the first hypothesis was wrong. The next hypothesis, personality. There is some literature that says that your ability to live long, healthy lives is in part dependent on personality factors. And so we were motivated by that to test this. And our hypothesis was the super seniors have a different personality profile than unhealthy individuals. And I'm going to talk about some of the preliminary data. The measurement tool that was used is called the NEO 5-factor inventory, and it measures 5 different aspects of personality. We use this because it is so widely used in the psychology field that there are literally thousands of publications that talk about college students, middle-aged people, all sorts of people whose personalities have been measured with this particular tool. And initially, we compared to the literature. So this study was done by Clarissa Gibson, who happens to be in the room here. I won't embarrass her, but you can look for her later based on her photo. And Clarissa finished her master's in my lab a couple of years ago. She had the very fun job of going and doing this personality questionnaire with the super seniors. She interviewed 42 supers in their homes, 27 men and 15 women. And they had aged a little bit since they were first recruited. This was a few years after their recruitment. And we compared them to a big paper published on Medicare patients in the United States. And compared to that literature, we found that the super seniors were much less neurotic. And neurotic, I tend to think of neurotic as a kind of a, it's going to go wrong, the world is against me kind of way of thinking. The super seniors are not like that. They are actually more extroverted and open and slightly agreeable and conscientious. The men, I believe, in particular, were a bit more extroverted. So conscientious makes sense in terms of health. Maybe these are the guys that eat their broccoli. They're conscientious. So that would make sense. So we do have to be careful with our interpretation of this as well, though, because people with this profile might be more likely to volunteer for research studies. So there can be a bias there. Personality, according to my reading of the literature, it was generally thought that personality is stable over a lifetime, but my colleagues who work in gerontology are disputing that later in life. They're not sure that it's absolutely stable. So does a personality profile of low neuroticism and higher extroversion and greater conscientiousness help you live longer. We're not sure, but it may. The third hypothesis was done by a method called exome sequencing. So here we're getting into a little bit of genome sequencing. If you were to sequence a genome, you would be putting together the whole ATGC sequence across all of the chromosomes. An exome is kind of a genome light. You pull the genes out and sequence only them, and so you're only sequencing about 1% of the genome, and it's a little faster, it's a little cheaper, and it's easier to analyze. So we did a pilot study of this, and to look at this, actually our hypothesis, this was my favorite hypothesis of all. So this hypothesis, the super seniors have fewer common genetic variants that have been associated with cancer, cardiovascular disease, pulmonary disease, dementia, and diabetes than regular people. So why do they not have these diseases? The hypothesis is, well, they don't have the genetic variants that cause those diseases, or they have fewer of them. That was our hunch. And so we did a very small study. We focused on the most exceptional super seniors of all, Robert Wiener and his older brother. So we took DNA from the brothers, and we sequenced their exomes, and we looked at genetic variants in the two brothers. So a very small study, just to kind of get started. So what we looked for was genetic variants that have been implicated in common complex diseases by research studies all over the world. And lucky for us, these are curated at the NIH in Bethesda, and they have a database of all of these studies accumulated. And so the data's all in one place. We could download a list of variants for each of these diseases, and we could say, are these variants in the two brothers? So that's what we did. So how many of you think that the two brothers, who are 107 and 109, and we're both super, how many of you think that they're going to have fewer genetic variants, disease variants? Okay, about half of you. Well, wrong again. And I was wrong too. I really thought this would be the case. So with the caveat, this is of course a very small study, the brothers had essentially exactly the same number of variants as regular people did. And there are other groups, research groups, who have done similar studies focused on different subsets of variants. They have mostly found findings that support our results as well. So it's fairly consistent, perhaps with the caveat that one group in the United States found that their healthy elderly, who were only 80 and older, were slightly lower in Alzheimer's disease-related variants and cardiovascular variants. But they did the study with whole genome sequencing, so it was a little bit different. And last, I'm going to show you work on one specific gene. My postdoctoral fellow, Julius Halashek-Wiener, did work on quite a number of candidate genes, and I'm not going to show them all today. My graduate student, Lauren Tyndale, who will be defending her PhD later this year, she did the work on ApoE that I'm going to describe. So ApoE is a gene that has been implicated in late-onset Alzheimer's disease. ApoE has three forms, and they're called Form 2, Form 3, Form 4. Form number 4 is associated with Alzheimer's disease and cardiovascular disease. And Form 2 is associated with longer life in previous studies. So ApoE4 is the most significant and consistent inverse association with longevity. And it's really the only association which you see in multiple different studies where it's consistent most but not all of the time. Other findings have been quite variable between studies, and we think they probably vary depending on the exact definition of health that's used, or perhaps the ethnicity or the part of the world, and so on. So ApoE is sort of the big one. Okay, so ApoE is a protein that relates to Alzheimer's disease because in Alzheimer's you have a protein called amyloid beta. and it's the buildup of amyloid beta essentially in aggregates that is part of the pathology of the disease. So ApoE has a role in removing amyloid beta, getting it across the blood-brain barrier, and the E4 form is less efficient at doing that, so is more likely to be correlated with Alzheimer's disease. So the hypothesis, the super seniors are less likely than controls to carry the E4 allele. How many of you agree with this hypothesis? Okay, you're scared of hypotheses now, aren't you? You've been wrong too. Don't be discouraged. It's how it works. Okay. Well, the hypothesis held up this time. And the data was actually very, very nice. So here, you can see, so what we do in this is called an association test, odds ratio. So the E3 form is the normal form. And so compared to the normal form, which we give a 1.0, what are your odds of being a super senior if you have if you carry one or more copies of the 4 form? Well, you're only 59% is likely to be a super senior. So you've taken your likelihood of being a super senior and cut it in half by having one or more copies of the ApoE4. We can look at this a little bit differently. We can break it down by exactly what combinations of the forms each person has. So everyone inherits one form from their mother, one form from their father. So they have 2 each. And the comparison is the E3, the normal form. We give that a 1. And compared to that, someone with 2 copies of the 2 form is 5 times more likely to be a super senior. So in our data, the E2 form really does look like it is associated with longer, healthier life. And conversely, if we look at individuals with the E4 form, 2 copies or 1 copy, people with 2 copies are only 35% as likely. So your odds of being a super senior are reduced. These are not all significant, but the trend is very as expected, and the overall p-value is significant. So we were very reassured by this because it also showed that we are recruiting properly. Now, one other way to think about this data, you can also say some super seniors do carry an ApoE4 allele, but they don't have Alzheimer's disease. So look down here. These guys, 4 people with 2 copies of E4 and 84 people with 1 copy of E4, and they are still super seniors. So what do you think this is due to? Think back to your human genetics lesson. What's this called? Penetrance. This is penetrance. They're carrying the disease allele, and they're not showing the disease. So what is it about these people that is protecting them against this disease? No, E4 doesn't always cause disease. It's not fully penetrant. I'm kind of in a circular argument there, but okay. So just to summarize the results of our hypotheses, the super seniors did not have longer telomeres than others their age, though their telomeres were clustered around a potential Goldilocks value. The super seniors had a personality profile that was different from less healthy seniors in the literature. 2 exceptional super senior brothers did not have fewer disease variants than other people. And ApoE4 is underrepresented in super seniors, and ApoE2 is overrepresented, exactly as we would have expected. And we have other candidate gene work that I won't go into today. So what are the genetic factors that cause healthy aging? If it seems from our work and from the literature that it's not an absence of bad genetic variation, is it presence of protective variants? You might be wondering, why is there a fly on that slide? That fly is a very nice specimen. In the fly, actually not this particular fly, but generally speaking. There is a phenomenon that has been observed in Drosophila, the fruit fly, called genetic buffering. And you can take a strain of fly that has a lot of genetic variants that cause problems. And in the fly, they focus on phenotypes that you can score by eye, looking at the flies under the microscope. So for the fly, it might have bulging eyebrows and wonky bristles and so on, things that are apparently not good if you're a fly. If you have a high expression of a certain gene and you introduce it into that fly, it masks all of that deleterious variation. Not just one of it, but all of it. So it is a one-on-many suppressor of negative genetic variation. So this is very well documented in the fly, and frankly, flies are just like humans, or humans are just like flies. If it's seen in the fly, why not in the human? And so this is what we're going after now. And so with some funding from the Canadian Cancer Society, we have a study where we are looking for genetic buffering in humans. And that's what we're currently working on. In all of our studies, larger sample size is always better. So we're always on the lookout for more super seniors. So we're welcoming new participants. If you are a super senior, if you have a super senior friend or a super senior grandparent, you're very welcome to give them our phone number or our study coordinator's email. I have some green sheets that we can pass out after as well that have this information. And I'd just like to acknowledge all the hard work, particularly of students over the last years. Everyone whose name is in red is a student who worked on the healthy aging study. And there would be another set of students who have worked on the lymphoma families study. So my collaborators, Joe Connors, Denise Daly, New Lee, John Spinelli, are gerontologists and clinical collaborators, and in particular, Julius Halashek-Wiener, Lauren Tyndale, and all other students. The work's been funded by the Canadian Cancer Society and the Lottie and John Hecht Foundation, and past support from CIHR, Canadian Institutes for Health Research, and the Michael Smith Foundation. So also a special thanks to members of the media who very generously have put our study phone number and email in their stories and read them over the air. And here's the lab doing typical lab activities. Thank you.
So just hand the mic back and forth, I guess. Well, wasn't that fascinating? What I love about the presentation is not only do we learn a lot about super seniors, but a whole bunch about research methodology and about how a researcher goes about asking questions, posing hypotheses, some of which prove to be correct, some of which prove not to be. And so we learn not only about the subject of the lecture being super seniors, but also about the methodology that produced the results and how excellent research is conducted. And I'm sure that has prompted on the part of some of you some questions or comments, and we have a bit of time for that. You may even end up on the YouTube channel if you so choose. Heather over here has a microphone. There's someone right over here with a hand up. So Heather's going to get lots of exercise and become a super senior by the process of doing so. Right back there. If you stand up and just say who you are and then ask a question, that would be really helpful.
Hi, my name's Marcia. I just want to ask, do you find in the general population there's an average of how many variants the average population carries, like 1, 2, 3, 4 variants?
Oh,
do they run really high or low? Like,
so this is on. So there are many variants. There are thousands of variants.
Yeah.
They tend to have effects that are very, very small. So you could have a variant that increases your risk of a specific disease by 1%, 20%, maybe doubles your risk, but most variants are in that range. You very seldom find a risk variant that is going to, actually, odds ratio of 5 is a big odds ratio. So they're mostly around 1.5 to 2. So we have many risk variants. You could say that we all have them. We have different ones. We may have slightly different numbers, We all have collections of bad variants. But it's naive to think of them as bad, because we're now realizing that what's bad for one disease might be good for another disease. And so the combination of your disease alleles and your lifestyle and your circumstances is what produces the overall effect.
I want to know, have you ever heard of the theory, Dr. Beer, he was a Scottish doctor in the early 1900s, his theory on cancer, that pancreatic enzymes, specifically trypsin, protein digesting enzymes, stop cancer and cure cancer?
I don't think that's probably not an actively applied treatment anymore.
Oh, okay.
Yeah.
And just briefly, I have the EPO4 gene.
Okay. Okay. Yeah. Well, you still might go on to be a super senior.
Thank you.
Okay, Shiraz has a question right up here about super grandparents, maybe.
Yeah, thank you. My question is about the seniors, the super seniors. Did they have a vision of living up to 100, or do right from when they were 40s or 50s, like thinking that I want to live 100, like me saying I want to live 100? And also, there is a Berlin study, I'm told, that said that seniors who take care, or grandparents who take care of grandchildren, tend to live longer because they feel they need to give their love to their grandchildren. Mm-hmm. I think it is a Berlin study. Mm-hmm. And yeah, so, and I find that a lot of youngsters, if I ask them if you had a choice, how long you want to live, often they say 80, and I ask them why, they say because they don't want to get Alzheimer's. Mm-hmm. Do you have any comment?
I knew there'd be a grandparent angle to this question, so.
Exactly. So the first part of your question, did the super seniors have a vision that they were, or an idea that they were going to be the ones to live long? Actually, I would say the ones that I've talked about that with, and Del Richards comes to mind. He was the exact opposite. He was absolutely surprised that he had lived that long. And the reason he was surprised is because his parents had died of cancer, and his sister died of cancer, and his first wife died of cancer. And so his question was, why haven't I died of cancer? And that was one of his motivations for joining the study. He was curious why he hadn't developed the disease. When you say that you had heard that grandparents who are involved in raising their grandchildren tend to be healthier, I haven't read that study, but the result doesn't surprise me. There's a lot of evidence that shows that the more active and engaged seniors are, and the more of a community of support they have around them, the more healthy they are. And you might think of this as the grandparents helping the grandchildren, but actually the grandchildren are also helping the grandparents. They're keeping them moving. I mean, keeping up with a child can be quite a tall order. So I can see easily why that's good for older people.
Changing diapers is a good strategy for aging. We have a question over here.
Hi. I was wondering why you're not including people who had cancer when they were younger, but are now 95 or 97 and are very healthy, because I'm not a scientist, but if we define healthy aging only by people who never had any of these things, aren't we limited in our definition of it? Because I'm thinking also of Olga, because I knew that when she died and she had autopsy, it revealed that she had either a cancer or tumor that she was able to do whatever she did with. Mm-hmm. But what I mean is that she was carrying a disease, but she was still extremely healthy. So
yeah, she was asymptomatic until her death. And actually, Olga and some other of our super seniors, anecdotally, they show a thing that is referred to by other gerontological researchers as compression of morbidity. That it's known that if you live to 100, you tend to be healthier in addition to long-lived. And when you pass away, you tend to pass away more suddenly. So your years of infirmity are shorter, and your years of health are longer. And so it's hard to separate the longevity and the healthy aging. But your question about why not include cancer survivors in the study, we did think about that. And we didn't do it because we felt that it's a different phenotype. And also, we also wanted to study the phenotype that in a sense is more desirable for the individual, not having to go through cancer, and the phenotype that's cheaper for the healthcare system as well. But that, in a way, it's a very interesting phenotype, but a different one. Mm-hmm.
Although you said on the radio today, just if you didn't catch it, that a lot of the super seniors have incurred serious stress
Yes.
in their lives of different kinds, not cancer, but other kinds of stress, and that doesn't seem to have been a contraindication to becoming a super senior.
No, it didn't. A lot of them have had very stressful lives, and they're still super.
There's a question right over here, Heather.
Did you determine how environment factored into promoting healthy aging?
No, we didn't. And it's because we didn't feel that we could quantitate that, asking them what their environment was early on. We decided to leave it to the cohort studies where people are followed over time. Actually, if people are interested in joining such a cohort study, the BC Generations Project would be the one to join.
The questions, one back here, gentleman back here, keeping Heather very busy here.
Thanks. Hi, I was just wondering if there were any diseases or conditions that super seniors tended to have that in your cohort that you'd arranged.
They do. They're not completely free of disease, and that was part of when we were thinking of how do we want to define a super senior. If we excluded on all diseases, we thought we'd end up with nobody in the study. We do find that quite a few of the super seniors, a certain number have macular degeneration, which becomes more prevalent as you're older. Osteoporosis is not uncommon in the super seniors. And some of them had high blood pressure. We didn't exclude on that because we didn't consider it a disease for the purposes of our study. It's sort of an intermediate phenotype, although it's usually medicated successfully.
But these are conditions or diseases that you didn't screen people out for, so they came into the study with them.
Yeah.
What about people who contract the diseases after they came into the study that weren't present before they came into the study? Is there anything to say about that, about super seniors? Are they more susceptible to some kinds of contracting some kind of diseases later in life than others?
They don't seem to be. We say once a super, always a super. So once you're in, we never, you're in unless you choose to withdraw. That's also fine. They do go on to develop some diseases, though. So, yeah.
All right, good to know. And there's a gentleman right here with his hand up. Where did Heather go? Over here. Okay. We'll take 2 or 3 more questions, and then we'll take our reception break. Yep.
I happen to have known Olga. She was a retired teacher in Burnaby, and she delighted in coming back to our retired teachers' luncheons and telling us what she was doing and really stimulating the people that were 30 years younger. But Olga told me that she did not get involved in track and field until she was 70 years old.
Yeah.
So what message does that send?
So that's a really interesting question. And she wasn't inactive early in life. Actually, Olga was very athletic as a child. She played all kinds of sports. She was a really little sport-oriented child. And then when she grew up and she had her children, she didn't do so many sports. And then when she was retired, she took up sports more again. And for a while, she was doing softball until one day she had a softball injury and she decided she was going to do something different that just involved doing it as an individual, not running into other people. I think I have that anecdote right. You'll have to read Bruce Grierson's book, but Yeah, it may be that people who take up sports later in life have fewer historical injuries. Although Robert Wiener is a case in the opposite direction. He kept on exercising regardless of his hockey injuries from his youth.
This gentleman right here, Heather. And then we'll do the last question back over here.
Porrick Slater, maybe a sequence or segue to the question on activity levels. I found it interesting that the super seniors had a BMI, which was on the sort of higher end of the range. Now, BMI has been talked about as having some flaws for athletic, muscular, rugby-type players, but my observation, maybe it's partly a question, is given that they had a higher BMI than you might have expected, might that suggest that they were not just doing aerobic conditioning, but also strength conditioning, which we're hearing more is important for people as they age?
Yeah, it's definitely very important. This doesn't come from our work, but from other people's work, that doing weight-bearing exercise is important for older people to maintain their muscle mass. Olga certainly did weights and did a variety of things as well. We didn't ask the super seniors if they do weights, so anecdotally I know that Olga did, but yeah, from other work you're correct.
Another hypothesis to be tested. And we'll take a last question from over here, a comment.
Hi, you guys. My name is Yasmin. I'm actually an undergraduate student here, and I don't see many students in the crowd. So this isn't a question, just a comment. So I figure skate, and every winter I'm always on ice skating. And this winter, the rink that I skate at said, all super seniors skate for free. And I thought to myself, I'm paying $5 every time I'm going in here. I want to be a super senior so I can skate for free. And I never saw any super seniors on ice. So hopefully we can get more people in, and especially if you said being more active prolongs your life too. So I mean, that's one of my life goals, to be a super senior and get to skate for free on the ice. I think that'd be cool.
The skating rink may have had a different definition of super senior. They're all super, perhaps, in that definition.
I was going to say, not only does that provide a great incentive for younger people, but it may provide a whole cohort for you to go and bring into the study. So
I'll be 200 by the time it finishes.
Well, it's great. What a fascinating work. I love coming to the President's Faculty Lectures because I always learn so much, and I know that the people who come have that opportunity too. So please join with me in thanking Angela Brooks Wilson for just a great lecture.